I'm a professor of biochemistry at the Hebrew University of Jerusalem, an adjunct professor at the Department of Microbiology in NUS. I work mainly on what is called signal transduction pathway, the biochemical pathways that connect the information from outside the cell into the cell and respond to whatever happens outside the cell. It's very complex. It involves enzyme called kinases, so this is my main expertise, the biochemistry, biology, and pathology. I think the MLID project is a very nice example of the activities that's going on here in CREATE, because it is a continuation of a previous project that was called MLID, Molecular Mechanism of Inflammatory Diseases. And I was part of that project too. And in this previous project, we were able to establish what we believe is unique and novel animal model for studying inflammatory diseases. And in the MLID, we are taking advantage of this model to study inflammation diseases in the liver, very specific. It can be used for studying inflammation in any organ, in any tissue in the body. This particular program is unique and specific, and the goal is to take the models that we established in the previous program and understand the mechanisms behind inflammatory diseases, specifically in the liver. This is a very big challenge. It is not known why large number of people suffer of inflammatory diseases in the liver, what's also called fatty liver diseases. About a quarter of the entire population of the world, billions of people, suffer of fatty liver. It is related to modern way of living, food consumption, but many people who keep healthy life develop this syndrome. And this syndrome by itself is not so bad, but huge percentage of them further develop more severe. It's called steatosis or liver cirrhosis, that further develop into cancer, hepatocellular carcinoma. So to understand how it starts, what is the molecular basis of the disease, this is the big challenge of MLID. And I think we are moving forward with this quite nice. Actually, our model deals already with a particular molecule, a kinase enzyme called T38. We know is active abnormally in many patients. I think this is the power or the sophistication of our model. Ferritin on and off in the liver and follow the exact event that it causes downstream when we activate it, and we can also monitor the liver recovery when we turn it off, which was quite unexpected and surprising, that when we activate this enzyme, after some time, many of the downstream targets of this enzyme are shut off. The expectation was that they are turned on because they depend on this enzyme. But what we are finding now, which is totally different than the dogma or the prevailing understanding of the current molecular basis of the disease, tissue, the liver respond to this activity, shut off downstream other enzymes and genes that everybody thought are going actually up, are going down. And probably part of the contribution of this enzyme to the development of the disease and to the suffering of patient is that many important targets are shutting off, not on. When we think about therapeutic approaches, we now should consider turning on some activity, not just inhibit with inhibitor drugs, etc. So we already know how to change our view of the molecular basis of the disease and to target differently the molecules that contribute to the development of the disease. Some of them should be reactivated or revived and not inhibited and shut off. So this, I think, is the main surprising contribution. And I already see some responses. It's a very good and strong collaboration. During MMID already, we had collaboration with 4, 5, later 6 groups within NUS. Some of them from the pharmaceutical, some from immunology, some from microbiology. We had a lot of discussion, co-experiments carrying out experiments together. We had 5 or 6 PhD students. So all this contributed very significantly to the establishment of the model. In MLID, I have also collaboration with 3 faculty members of NUS. So MLID is coming to an end. I think that we established nice connections between HUJ and NUS that hopefully will continue beyond the time of the project. Using this model that we established in MLID, we obtain tons of data. We now know exactly what happens in the liver when we induce the activity of this enzyme TSRP8. All changes in thousands of lipid molecules. The changes is going up or down in gene expression, protein phosphorylation. So analysis of these tons of data and translating them to targeting molecule for therapy will obviously continue. You cannot just have this data, obtain them, pick up the major observations and stop. Yes, I'm not going to give it up. So obviously we are going to continue, yeah.